Hey everyone, and welcome to another deep dive. Have you ever looked out at a forest or even just a single plant and wondered how they make their own food?
Yeah, it's really quite remarkable.
But today we're going to be diving into the fascinating world of photosynthesis, going way beyond the basic stuff you learned back in school.
Definitely. It's a topic with so much depth, and, it's easy to kind of just gloss over the surface.
Totally. But we're going deep. And we got a bunch of really cool excerpts that you guys sent in.
Yeah, some really fascinating perspectives.
So our mission today is to figure out the nitty-gritty of how plants create food, the who's who of this whole process, and also what things can help or hinder it.
Absolutely. It's way more complex than most people realize. We all know plants need sunlight.
Right. That's kind of photosynthesis 101.
Exactly. But have you ever stopped to think, where does that energy actually go?
That's a good question. I mean, I know we eat plants for energy.
Right. And that's the amazing part. Plants, they're what we call autotrophs, meaning they can produce their own food.
Oh, yeah, I remember that term.
So they do this by converting light energy from the sun into chemical energy, which is then stored in the form of sugars.
So sunlight is essentially transformed into plant food.
Pretty much. And then as a bonus, they release oxygen into the atmosphere as a byproduct.
Wow, so photosynthesis is kind of the reason we're all here.
You could say that. It's the basis of almost all life on Earth. Provides us with both the food we eat and the oxygen we breathe.
Okay, yeah, that's definitely deep dive worthy. But I'm curious, how does it actually work? Where does this whole magical transformation take place?
Well, the magic happens inside these tiny structures within plant cells called chloroplasts.
Ah, the chloroplasts.
Yep, think of them like mini factories, dedicated to harnessing light energy.
Okay, so that's where chlorophyll is, right? Which gives plants their green color.
Exactly. Chlorophyll is the superstar of photosynthesis, but it's not a one-man show.
Oh, so there's more to it than just chlorophyll.
Way more. Chloroplasts are these complex structures with different compartments, each playing a specific role. You've got the grana, which are stacks of these membrane-bound sacs called thylakoids, and that's where the light energy is captured and converted into chemical energy.
So the granas are kind of like the solar panels of the plant cell.
I like that analogy. Then you have the stroma lamellae, which connect the grana and basically help transfer energy between them.
Okay.
And finally, the stroma, which is the fluid-filled space surrounding the grana, and that's where the actual sugar synthesis takes place.
Wow, so it's like a well-organized factory with different departments working together to create the final product.
Exactly. Each part has its own important job to do.
But we see so many shades of green and even other colors in leaves. So is chlorophyll the only pigment at work here?
It's the most famous one for sure, but plants actually use a whole variety of pigments, each absorbing light at slightly different wavelengths.
Oh, interesting.
Yeah, this allows them to capture a wider range of light energy, kind of maximizing their intake.
Makes sense. So it's not just about being green. There's a whole rainbow of pigments contributing.
Exactly. So we've got chlorophyll A, which is the main pigment that captures light energy. And then you have these accessory pigments like chlorophyll B, xanthophylls, and carotenoids. And they kind of act like a support system. They absorb light at wavelengths that chlorophyll emisses and transfer that energy over.
So it's chlorophyll V is the lead singer, and the accessory pigments are the backup band, making sure all the light energy gets used.
Perfect analogy.
That's awesome. So these pigments are busy absorbing light, but how does that actually translate into usable energy for the plant? What's happening at the molecular level?
Well, it's a bit complicated, but basically when a pigment molecule absorbs light energy, it excites an electron, kind of boosts it to a higher energy level.
Oh, okay. So it's giving that electron a jolt of caffeine.
Yeah, something like that. And then this excited electron triggers a whole chain of reactions that ultimately lead to the conversion of light energy into chemical energy.
Okay, so light hits the pigment, electrons get energized, and then what? How does that actually make food for the plant?
Good question. So photosynthesis actually occurs in two main stages. You've got the light-dependent reactions, which obviously require light, and the light-independent reactions, sometimes called the Calvin cycle, which can actually occur without light.
Ah, okay. So it's a two-part process, kind of like a relay race. The light reactions capture the energy and then pass the baton to the carbon-fixing reactions.
Exactly. The light reactions capture light energy and convert it into chemical energy in the form of ATP and NADPH. And these molecules then power the Calvin cycle, where carbon dioxide is fixed into sugars.
Okay, so let's take a closer look at these light reactions first. Where do they take place?
They occur in those thylakoid membranes we talked about earlier, the ones stacked up in the grana. And these membranes are packed with photosystems, which are these complexes of proteins and pigments that work together to capture light energy.
So we're back to those solar panel grana. What's going on inside these photosystems?
Well, there are actually two types of photosystems, cleverly named photosystem I and photosystem II.
Okay.
And they work together to transfer electrons from water to NADP, plus generating ATP and NADPH in the process.
Photosystem I and II, are they named in the order they work in the process?
Actually, no. They were named in the order they were discovered, which can be a little confusing, you know? But the key is that both photosystems are essential for capturing light energy and driving this electron transport.
Okay, so it's more of a carefully choreographed dance than a straightforward relay race. Can you walk me through the steps of this dance? Where do the electrons come from and where do they go?
Sure, it all starts with photosystem 2. When a photon of light hits photosystem 2, it excites an electron in the reaction center chlorophyll, and this excited electron is then passed along an electron transport chain, similar to what happens in cellular respiration.
Oh yeah, another connection back to high school biology.
Right. And as the electrons move down this chain, their energy is used to pump protons across the thylakoid membrane. And this creates a proton gradient, which is then used to generate ATP through a process called chemiosmosis.
This is like those energized electrons are powering a tiny pump.
Exactly. And as the electrons continue down the chain, they eventually reach photosystem 1. Another photon of light excites another electron, which is then passed to NADP plus O-Phe, reducing it to NADPH.
Okay, so we've got electrons flowing from water to photosystem 2, down the chain to photosystem I, and finally to NADP plus O-Phe, creating ATP and NADPH along the way. But you mentioned water splitting earlier. How does that fit into all of this?
Ah, yes, that's crucial. So for this whole electron transport chain to keep running, photosystem 2 needs to replace the electrons that it loses, right? It does this by splitting water molecules into oxygen, protons, and electrons.
Wait, so the oxygen we breathe comes from water molecules being split in plants.
You got it.
Whoa, mind blown.
It's amazing, isn't it? So the electrons from water replenish photosystem 2, keeping the process going. The protons contribute to that proton gradient for ATP synthesis, and the oxygen is released as a byproduct.
Wow, it's like nature's ultimate recycling program, using water to generate both energy and oxygen. But hold on a second. We haven't talked about cyclic and non-cyclic photophosphorylation yet. What's the difference between those?
Good catch. So what we've been talking about is non-cyclic photophosphorylation, which involves both photosystems I and A to produce both ATP and NADPH, but sometimes only photosystem I is involved, creating a cyclic electron flow where electrons are cycled back to photosystem I instead of being used to reduce NADP plus phosphorylation. And this is called cyclic photophosphorylation.
Oh, I see.
And it only generates ATP, not NADPH.
So it's like the plant cell has different modes depending on its needs.
Exactly. It can regulate which pathway is active based on the light conditions and the demand for ATP and NADPH.
That's pretty cool.
Right. It's super dynamic.
This is incredible. So much is happening at like the molecular level in plants.
Right. But we haven't even touched on the Calvin cycle yet, where all that ATP and NADPH is put to work to actually build those sugars from CO2.
Okay. I'm ready to hear about that sugar factory.
Yeah.
Shall we move on to the Calvin cycle?
Let's do it. Okay, so now we're stepping into the sugar factory, otherwise known as the Calvin cycle.
Ooh, I like that. The sugar factory. So we've got all this energy from the light reactions, the ATP and the NADPH, just waiting to be put to use. What happens next?
Well, now we move to the carbon fixing stage, where carbon dioxide from the atmosphere is transformed into those energy-rich sugars.
Okay.
And this all happens in the stroma of the chloroplast. Remember that fluid-filled space surrounding the grana?
Oh, yeah, right.
So we're moving from the solar panels of the grana to like the factory floor of the stroma, where the real production happens. The Calvin cycle is basically a series of enzyme-catalyzed reactions.
Okay.
And we can break it down into three main stages: carboxylation, reduction, and regeneration.
Carboxylation, reduction, regeneration. Got it.
Yeah.
Let's start with carboxylation. What's going on there?
So carboxylation is the first step where carbon dioxide is captured and incorporated into an organic molecule.
Okay.
And the key player here is an enzyme called ribulose 1-phenol bisphosphate carboxylase oxygenase, or Rubisco for short.
Rubisco. I've heard of that. Isn't that like the most abundant enzyme on Earth or something?
You're right, it is. It's incredibly important because it catalyzes the reaction between carbon dioxide and a 5-carbon sugar called ribulose 1-phenol bisphosphate, or RuBP. And this reaction produces two molecules of a 3-carbon compound called 3-phosphoglycerate, 3-PGA.
Okay, so Rubisco grabs CO2 from the air, attaches it to RoBP, creates 3-PGA.
Got it. Then what?
It becomes reduction,
Right.
where 3-PGA is converted into glyceraldehyde 3-phosphate, or G3P, and G3P is a precursor of glucose and other sugars.
And this is where the energy from ATP and the reducing power of NADPH, both generated in the light reactions, come into play.
Ah, so this is where those products from the light reactions finally get used.
Exactly.
What do they do?
Well, ATP provides the energy needed to drive these reactions forward, and NADPH provides the electrons to reduce 3-PGA to G3P.
So ATP and NADPH are like the fuel and the building materials for this sugar construction project.
I like that. A perfect analogy.
Thanks.
Now, not all the G3P produced goes directly to making sugars.
Oh,
Some of it is needed for the third stage regeneration.
regeneration sounds important.
It is. It's essential for keeping the whole cycle going. So in the regeneration stage, some of the G3P is used to regenerate RoBP, that 5-carbon sugar we talked about earlier, the one that acts as the starting point for grabbing more CO2. And this ensures that there's a constant supply of RoBP to keep the cycle running.
So it's like a self-sustaining sugar-producing machine.
Exactly. It's pretty remarkable when you think about it. The Calvin cycle is a real feat of biochemical engineering.
Yeah, it's incredible. So we've been talking about what's called the C3 pathway, right?
Right.
Which is the most common type of carbon fixation.
Mm-hmm.
But you mentioned a C4 pathway earlier. What's that all about?
Yes, the C4 pathway. So that's an alternative way that some plants fix carbon, particularly those that have adapted to hot, dry environments.
Oh, okay. So how does the C4 pathway differ from the C3 pathway?
So in C4 plants, the first product of carbon fixation isn't a 3-carbon molecule like 3-PGA, but a 4-carbon compound called oxaloacetate,
Okay.
hence the name C4 pathway.
Right.
This reaction is catalyzed by an enzyme called PEP carboxylase, which has a higher affinity for CO2 than Rubisco does.
So it's like C4 plants have a more efficient way of grabbing CO2 from the air.
Yes, exactly.
Does that give them an advantage?
It definitely can, especially in those hot, dry environments where plants need to conserve water by closing their stomata.
Right, which would limit CO2 intake,
That's-
which would be a problem for C3 plants.
Exactly. So C4 plants, they have this clever workaround. They've evolved a special leaf anatomy called Cran's anatomy.
Cran's anatomy?
Cran's anatomy, yeah. So think of it this way. The Calvin cycle is confined to bundle sheath cells surrounding the leaf veins,
Okay.
like an inner sanctum.
Right.
And these cells are surrounded by mesophyll cells, where PEP carboxylase is busy fixing CO2 into oxaloacetate.
Okay, so it's a two-step process. CO2 is captured in the outer mesophyll cells and then transported to the inner bundle sheath cells where the Calvin cycle happens.
Exactly.
Right. And this spatial separation is what allows C4 plants to concentrate CO2 in those bundle sheath cells, which boosts the efficiency of Rubisco and minimizes a process called photorespiration.
Photorespiration, what's that?
So think of photorespiration as like a hiccup in the system. It happens when Rubisco, instead of fixing CO2, reacts with oxygen. And this is wasteful and reduces the efficiency of photosynthesis.
Oh, so it's like Rubisco grabs the wrong molecule sometimes.
Yeah, pretty much. And, Rubisco evolved when the atmosphere had much lower oxygen levels.
Oh, okay.
So it wasn't a big issue back then.
Makes sense.
But today, with higher oxygen levels, photorespiration can be a significant drain on energy for those C3 plants.
But you said C4 plants have found a way to minimize photorespiration. How did they do that?
Right. So by concentrating CO2 in those bundle sheath cells, C4 plants create an environment where Rubisco is much more likely to encounter and fix CO2 instead of oxygen.
It's like they've optimized the working conditions for Rubisco, making sure it has a steady supply of its preferred material.
Exactly. So you could say that C4 plants are like these super efficient, drought-tolerant champions of photosynthesis.
Yeah, that's awesome. I mean, who knew so much complexity and ingenuity was packed into a tiny leaf?
Right. And we've only just scratched the surface. We haven't even talked about all the environmental factors that can influence photosynthesis, like light, temperature, CO2 levels, water availability.
Right.
I'm ready to dive deeper into those factors. How do they impact this whole incredible process?
Right.
Okay, so let's talk about those environmental factors. I mean, light has got to be a big one, right? Since it's like the ultimate energy source for this whole process.
Oh, absolutely. Light intensity, the quality of the light, how long the exposure is, all of that influences how efficiently a plant can photosynthesize. Generally speaking, more light equals a higher rate of photosynthesis. But there is a catch.
A catch? What do you mean?
Well, it only works up to a certain point.
Oh, okay, so it's like you're giving a plant more sunlight, it's like giving it more fuel, but why wouldn't more always be better?
Well, every plant has what's called a saturation point,
Okay.
and beyond a certain light intensity, the rate of photosynthesis plateaus even if you keep increasing the light. It's like the plant's photosynthetic machinery hits its maximum capacity. It can only process so much light at once.
Oh, so it's like overloading a circuit. Too much of a good thing.
Exactly. And in extreme cases, excessive light can actually damage the photosynthetic machinery,
Oh, wow.
leading to a decrease in efficiency.
Interesting. Okay, so light is key, but there are limits.
Huh.
What about carbon dioxide? I mean, that's one of the raw materials for making sugars in photosynthesis, right?
Absolutely. Carbon dioxide is essential for that Calvin cycle we talked about, and its concentration in the atmosphere can directly impact the rate of photosynthesis. In fact, increasing CO2 levels can sometimes lead to a boost in photosynthesis, particularly for C3 plants.
Oh, really? So with rising CO2 levels because of, climate change, could some plants actually benefit?
Well, it's a complex issue. While some plants might initially see enhanced growth with increased CO2, there are potential downsides as well.
Okay.
For instance, altering CO2 levels can change the composition of plants and their nutritional content,
Oh, wow.
which can then impact herbivores and, the whole food web.
Right, the ripple effect.
Exactly.
Right.
It highlights how interconnected everything is within an ecosystem.
Yeah, totally. Okay, so what about temperature? I know enzymes are very sensitive to temperature changes, and photosynthesis relies on like a whole bunch of enzymes.
Yeah. Temperature is a critical factor for sure. Photosynthesis, like all enzymatic reactions, has an optimal temperature range where it operates most efficiently. If it gets too cold or too hot, the rate of photosynthesis will decline.
Right, right. And I'm guessing that optimal temperature range varies depending on like the plant species and where it lives, right?
Exactly. A tropical plant, for example, will have a much higher temperature optimum than a plant living in a colder climate.
Makes sense.
Yeah.
Okay, last but not least, water. We know it's a reactant in photosynthesis, but how does its availability impact the process?
Well, water stress can significantly limit photosynthesis. When a plant doesn't have enough water, it closes its stomata to prevent water loss.
Right, to conserve water.
Exactly. But this also restricts CO2 intake, which we know is essential for the Calvin cycle.
Ah, so it's a trade-off. The plant tries to conserve water, but ends up limiting its ability to photosynthesize.
Right. Plus, water stress can lead to wilting, which reduces the leaf surface area exposed to light. So that further impacts photosynthesis.
Wow. It's amazing how interconnected it all is. So we've got light, CO2, temperature, and water. All these factors influencing photosynthesis in different ways. How do scientists even begin to make sense of this whole complex interplay?
Well, there's this helpful concept called Blackman's law of limiting factors.
Away.
And it basically states that the rate of a process like photosynthesis is limited by whichever factor is in shortest supply.
So it's like a chain is only as strong as its weakest link.
Exactly. A plant could have abundant light and ideal temperatures, but if CO2 levels are low, that's going to be the bottleneck. It's going to limit the rate of photosynthesis.
This is so fascinating. It really is like a puzzle where all the pieces have to fit just right for a plant to thrive.
It is. And understanding these factors is not only crucial for understanding plant biology, but also, for things like agriculture and ensuring food security.
Yeah, absolutely. Well, we've journeyed from the microscopic world of chloroplasts to the global implications of photosynthesis. What are some final thoughts for our listeners as we wrap up this deep dive?
I think it's important to remember that photosynthesis isn't just something happening in a lab or a textbook. It's happening all around us, in every green leaf we see.
Yeah.
It's the foundation for the oxygen we breathe and the food we eat. And understanding this process can help us appreciate, the incredible interconnectedness of life on Earth.
That's beautiful. I think we all owe a huge debt of gratitude to plants and the amazing process of photosynthesis. Well, that concludes our deep dive into this essential and fascinating process. We hope you've enjoyed the journey and learned something new along the way.
Until next time.